Hello, welcome to the last lecture of the semester. Yay, you're almost there. Way to go for persevering, working hard. So yeah, this is chapter 15. This is the last chapter. This and the previous chapter 14 will be on this week's mini exam. In terms of assignments, there will be a mini exam for these last two chapters. We have on Tuesday, I assigned the last lab of the semester, so double check when those are due. And I am working on grading your lab reports. There are a lot of them. It's been a little crazy, so hopefully you should start seeing those. I'm planning on finishing them up during finals week. Right, so I thought I might share a little bit about this room I've been in lately. Um, so the Fighting Sioux decoration is on my left. That is the former mascot of where I went to college, University of North Dakota, where my husband went as well, and he is a massive hockey fan, so that college team is pretty well known, and now he cheers for the St. Louis Blues. So if you know any hockey fans, there's not a lot out here in Utah, send them my way. And then I have a friend who's an artist, and she painted that for us. That's a dragon who's kind of like climbing through the walls. Kind of fun. I use this room for my artwork. So I got an easel over here, also cat room, got cat tree, and the lovely kitty litter box. So when I've been teaching down here, I light a few candles, and that helps make it a little more manageable. So let's talk about genes and how they work. Right, so this chapter really brings everything full circle. Right, we're going to talk about the central dogma. This is this idea or this flow of information that is shared among all species, whether you are a fly or a human or a plant. We are going to talk about transcription and translation. So those are the two main components, the two main processes in the central dogma, and you should be able to understand what happens during each of these, in what order they occur, what products are made from them, and what the products are used for. So transcription is going to be the creation of RNA from DNA, and you should understand that there are some modifications that must be made to this RNA before it can move out of the nucleus to the cytosol where it undergoes translation. Translation is the synthesis of protein. So any protein in the cell that has to be made is made by translation. And this occurs in the cytoplasm, as I said before, at an organelle called the ribosome. So you're going to learn about the different steps of translation and where it happens in the ribosome. And then lastly, we're going to talk about how mutations in genes can lead to different proteins and how these different proteins can lead to changes in phenotype, so appearance or behavior or function in an organism. So first, let's talk about the central dogma. Right? So taking a step back, remember when we talked about the evolution of the horse? Yeah. So we saw that over time it got larger in size, longer legs, lost some toes, right? And also the teeth became flatter and adapted for grinding grass, right? So these are all different types of physical characteristics that changed over time. And they're all due to changes at a cellular level, right? If you're going to grow bigger, you need more cell proliferation, so more cell division has to happen. If you are going to develop bone in a certain shape, right? There's proteins that are responsible for developing and shaping bone, making its structure. And so a lot of the actions that cells perform are mediated by proteins or are performed by proteins. And so we know that genes or DNA is what determines what contains the instructions for making proteins, right? Genes are like our template, right, or a blueprint for building a house, for example, right? And so changes in that blueprint could result in an updated house structure, right? Changes in DNA can lead to differences in these cells due to changes in proteins, how they function, and the resulting changes in these cells can result in changes in the organism, right, in the phenotype. So we're focusing today really on the genetic changes. For example, what types of genetic changes could have allowed a horse to change its tooth shape, right, or to grow longer legs. Another example here we have are in our lizard, anole lizards. And if you watched the video, hopefully, from lab 11, about anole lizards, you learned about dewlaps. And here we have some different anole lizards with different colored dewlaps. And these are used for mating. And the brighter the color in contrast to the surroundings, the more attractive that male is to females. It's noticeable, right? So there are genes that code for the proteins that form these different colors, right? So the formation of these colors are pigments called pterin and carotenoid pigments, right? And so again, there are proteins, enzymes that are responsible for synthesizing these pigments. How these enzymes work determines whether you're going to have a green or a yellow or a red dewlap, right? And so these genes, there are genes that code for these proteins, right? And so if, for example, it is beneficial to have a red dewlap, the genes that coded for those enzymes that produced red dewlaps are the ones that get conserved, the ones that get passed down to offspring, right? So we're going to learn more about how these genes lead to phenotypes. So if there's a mutation in the gene that's responsible for synthesizing those pigments, could it result ultimately in a different colored dewlap that could maybe be beneficial? So again, what we're talking about here when we're talking about genes and proteins is the central dogma. And the central dogma, first described by Francis Crick, is that we have this flow of information where your DNA is coding for RNA and RNA codes for protein. And so the central dogma, when you say coding for, right, DNA is used to make RNA. That process is called transcription. And making RNA into protein or using RNA to make protein is called translation. Now, the way I remember the order, transcription and translation, transcription has a C in it, translation has an L, C comes before L in the alphabet. And I've used that trick for a very long time. It's a pretty good one. Do whatever you need to do, but you should know the central dogma. You should know the steps and those processes that convert DNA to RNA to protein. Here we have a more graphical representation of the central dogma. So again, this happens in all cell types, whether you're a bacteria or a mammalian cell, right? So DNA is made into RNA in a process called transcription, and RNA is then used to make protein in a process called translation. Notice that DNA and RNA look pretty similar to each other. They're both nucleic acids, whereas protein is a completely different type of macromolecule, right? Proteins are made out of amino acids. So another way to think about the central dogma is what these words transcription and translation literally mean. So transcription, a transcriptionist is someone who just writes down what someone is saying, right? So we wish we were all so lucky, right, when we're writing papers that we could have someone sit and type up what we say. That process is called transcription, right? Now, conversely, translation, we probably all know what that means. That's where you're converting one language into another, right? So a translator would help me to speak to someone in, I don't know, Spanish, though technically I should know how because I minored in it, right? But what a translator does is they're taking information and converting it from one form into another. So when you are doing transcription, DNA is being made into RNA, or is being used to synthesize RNA. That's just taking one nucleic acid sequence and turning it into another, using it to make another nucleic acid sequence. Just like a transcriptionist, if I'm talking in English, transcriptionist is writing down my words in English. But translation is changing it from a nucleic acid sequence to an amino acid sequence, right? So it's like you're shifting languages like a translator would do. Right? So that's another way to remember what's going on in the central dogma and in what order it occurs. So briefly, just kind of looking over what happens during transcription. So a gene, a DNA sequence in the genome is being told it needs to synthesize and needs to make its protein product, right? So transcription must first occur before then translation can happen, right? So to make a protein from a gene, first you have to have transcription, and that makes a message RNA sequence. And then you have translation where that message RNA sequence is used to make the final product, the protein. Now DNA is that template, right? It's the blueprint. RNA is a single strand complement of DNA. So remember, we've got our complementary base pairing A, T, C, and G. Well, in RNA, instead of thymine, instead of the T, we have uracil. So the complement to A, instead of T, in RNA, the complement of A is a U. And this message RNA is used, it's called message RNA because it's being used to make protein. There's other types of RNA, like tRNA and rRNA that we'll talk about later on, and they aren't used to make proteins. So message RNA is what we're focusing on here. The message RNA acts as a template as well to guide the synthesis of proteins, which are chains of amino acids. Again, this takes place outside of the nucleus at ribosomes. And this is when you're going to see some other types of RNAs, molecules being involved. So let's break down transcription in more detail. Here we have some DNA, genomic DNA in blue, and we've got a gene here that is told it needs to be used to make protein. So the first thing that's going to happen is transcription, where the message RNA is going to be made. And the message RNA is shown in red. It is complementary to the genome, the genomic DNA. You can see the genomic DNA at a more magnified view is in blue. And the sequence that actually results in coding for protein is called the coding sequence. And we have a region upstream, right, before the coding sequence called the promoter. And that's important for regulating whether or not this gene is going to be transcribed or not. And on the other end, we have a termination sequence, and that is what helps to tell enzymes this is the end of the gene, stop making RNA, right? NTPs, nucleotide triphosphates, just like when we make DNA, we use nucleotide triphosphates. When we make RNA, we use nucleotide triphosphates. So we're going to focus on eukaryotic transcription today. We have a variety of RNA polymerases in eukaryotes. We're just going to focus on RNA polymerase II. So just like when we are copying DNA, we have a DNA polymerase. When we are making message RNA, we have an RNA polymerase. And it makes a sequence that's complementary to that of the DNA that it is bound to. Now, the transcription has, just like with replication, an initiation, elongation, and termination phase. And initiation is going to happen at this promoter region. Again, this is upstream of the coding sequence. And what happens here is before the RNA transcript is made, proteins will come in. If the cell gets a signal that it needs to transcribe this gene, proteins will travel to the nucleus and bind to that promoter, and they are useful for telling the RNA polymerase, hey, come over here, this is the gene that we need to transcribe. Right? So that's what the promoter is important for. We're going to look a little bit more in detail at initiation of transcription. So we have transcription factors shown in blue, purple, and pink, right? So eukaryotic transcription, there's always lots of people and lots of proteins involved, right? And what I would like you to note, first of all, is that there's a transcription factor that helps to initiate everything. And the sequence, you're saying, how does the transcription factor know where to go? The first transcription factor that comes in binds to a sequence of DNA called TA with the sequence TATA. All right, so in that promoter region, there's a sequence of DNA that is TATA, and that is where the transcription factor knows to bind. So the transcription factor, the cell gets a signal that it needs to synthesize protein, it needs to transcribe this DNA. It comes in and it binds at that TATA sequence, recruits other transcription factors, and that complex tells RNA polymerase where to come and in what direction to start synthesizing RNA and so forth. Right? Now, I'm not going to play this video because they never work for me, but I encourage you to stop this, pull up the video, and watch it. It's a great overview of the central dogma of transcription and of translation. So let's work on a problem. You're given a DNA sequence and you need to determine the complementary RNA sequence. So think about your base pairing rules. All right, pause the video, try and figure out what the RNA sequence would be. So here we have a complementary sequence, right? A pairs with T, G pairs with C. I'd like you to also note that we have this anti-parallel system going on, right? The 3' end is pairing with the 5' end, right? This happens whether you've got two strands of DNA together or whether you've got one strand of DNA and one strand of RNA. They're both nucleic acids. They both follow that anti-parallel system. You may say, wait a minute, there's something wrong, right? So messenger RNA in general has something different than DNA. And that is that instead of thymine, there is the base uracil. So the actual correct answer is shown here, where we have the thymines, the T's are placed with U. Let's move on. So the message RNA has been made, all right? It's a single strand, and it's going to exit the nucleus to move on to translation, right, to guide the synthesis of protein. But before message RNA can leave the nucleus, there's some modifications that have to be made. And again, this is specific to eukaryotes. Prokaryotes, they don't even have a nuclear envelope. So there's no requirement of leaving the nucleus. It's already out there in the cytoplasm, right? So this is eukaryotic processes that must happen. So we call this RNA when it's first made precursor mRNA, right? It's not quite ready yet. So there's a few things that have to happen. First, there is a cap that's added to the 5' end, and this is providing protection for this RNA sequence, and it's also helping with the initiation of translation. So when translation occurs at the ribosome, it helps the ribosome know that this is a sequence that needs to be translated. There is also a poly A tail added to the 3' end. So the opposite end of that RNA strand just gets a bunch of A's added on to the end of it. And this is also a way of protection. It's kind of like a bumper on a car. If that gets damaged, it's okay because the important sequence, the coding sequence, is not affected. The last thing that has to happen to this message RNA is that non-coding sequences have to be removed. And so I'm going to show what that means on the next slide. With this precursor mRNA, is modified by a spliceosome. Right, so this is called splicing. Here we have the message RNA that has now had a 5' cap added onto it. Right, you can see it's got, you don't need to memorize the structure of it, but you should just know that it's on the 5' end. And the message RNA on the other end, the 3' end, has a poly A tail added onto it. Okay. Now when we talk about intron splicing, here we have a different cartoon demonstrating a gene. This time we have the promoter shows as kind of a blue circle, and we've got our coding sequence is made up of exons and introns. Right? So this is just a long sequence of DNA, and that sequence can be categorized into regions called exons and introns. And the exons are not needed, sorry, the introns are not needed to actually code for protein. So these need to be removed. So you can see during splicing that the intron sequences are cut out, and then the exons are pasted back together again by the spliceosome. Okay? And you may say, why have those introns in the first place? So what this does is this allows for some variety in the types of genes that the types of proteins that a gene can code for. For example, you can have where in addition to all the introns, say exon 2 also gets removed. So now you're making a protein with the sequence exon 1, 3, and 4. And that protein could have a different function and structure than the original protein that had all 4 of them. Right, so it allows for more diversity in the proteins that get made by one single gene. That helps to cut down on the size of the genome, which is already pretty large, right? So the important thing I want you to know is what introns and exons are, which ones get cleaved out, what is this process called, what enzyme is responsible for that. Right, so here we have another video that talks about transcription and translation that I would like you to stop and check this video out. Once you've done that, you can catch up with me on the next slide. Let's talk about what happens during translation in more detail. All right, so just as a reminder, translation, we are translating, right, a nucleic acid into a protein. In other words, nucleotides are being used to code for the synthesis of a polymer composed of amino acid subunits. And so we're taking this language of A's and T's and G's, or A's and U's and C's and G's, and we're turning it into lysine, amino acid, serine, valine, etc. Right? So how do you go from a sequence that contain 4 bases or letters in a variety of different combinations to how does that sequence code for the lysine amino acid or the valine amino acid, right? What is the secret code, right? So Marshall Nirenberg really helped to identify the genetic code. And what he found was that a sequence of 3 bases will code for 1 amino acid. Right? So for example, here we have AUG. I've just, in the second row, I've kind of separated out this sequence of message RNA into its codons. Right? AUG, that sequence of 3 nucleotides, codes for the amino acid methionine. And the next sequence of 3 nucleotides, ACU, codes for the amino acid threonine. So each set of 3 nucleotides is called a codon. And you'll see in your book that we have the genetic code all laid out. You can see, for example, that UUU codes for phenylalanine, and you do not need to memorize this. Right? So if you're given any questions on the mini exam about sequences and what they code for, you will be given this table. So another thing I'd like to note is that you can see that there is not only one codon for one amino acid, right? So for example, for serine, there are 4 different codons that will all code for the amino acid serine. Another thing you can notice in that table is that there are some codons that are called stop and start codons. So in order for the ribosome to know where to start making protein, just like we need to have a TATA sequence for the transcription factors to tell RNA polymerase where to go during transcription, during translation, there's a sequence in the RNA called that is AUG that tells the ribosome, oh, here's where I'm supposed to start making protein. Right, so the first amino acid in any protein is going to be methionine. So methionine is the amino acid that is coded by AUG, which tells the ribosome to start making the protein. And then the ribosome knows to stop synthesizing protein when it gets to stop codons, which could be a sequence of UAA, UGA, or UAG. Now, again, I said that there are multiple codons that can code for a single amino acid. And this means that the code is degenerate, right? So there's multiple ways to spell the same thing. Let's do a problem. Determine the amino acid sequence for the following message RNA. Right, so the first thing you need to do is break it down into its codons. And you'll notice the first 3 amino acids, the first 3 nucleotides, the first codon is AUG. And when you look through the table, you'll find that AUG codes for methionine. That is our start codon. The next set of 3 nucleotides, UAU, you look through the table and you find that codes for tyrosine. Next, we have AUU and GAU. Right? So you can find lots of quizzes online with problems like these to use as you're studying. Let's do another one. Determine the amino acid sequence from the following DNA. And so take a minute, pause the video, try and do this one on your own. Now, I'd like to point something out as we begin this problem. This is a DNA sequence, right? So what that means is that you, as long as you know the genome sequence for an organism, you can determine, just like at your table, right, at your desk, what the protein sequence is going to be, which is pretty cool. So here we have the DNA. DNA has to first be transcribed before it can be translated. So we have to find the message RNA for this sequence. So I've just broken it down, spaced it out to make it a little bit easier. So the message RNA, it's the complement of the DNA, and we're replacing the T's with U's. All right, so now we have our message RNA sequence, and now we can go to the genetic code to this table and find out what proteins it would code for. Again, you see that AUG, methionine, is the first amino acid in this sequence. So be very careful when you are doing problems to check, are you looking at an RNA sequence or a DNA sequence? Right? Now, if you remember, I said if you know the genome, the sequence of the genome, so if you have sequenced, say, the gene of a jellyfish or of a pig or of a mouse, you can figure out what the protein is going to be, what the sequence that protein, resulting protein, will have. The other thing that's really cool is that this code is universal. So the same AUG that codes for methionine in jellyfish also codes for methionine in pigs in humans, right? So this code is universal, and that is why we've been able to manipulate the genome, because we know, we can predict what's going to happen, because we know what proteins, what amino acids are going to be made from what genes. And so I can take the gene that is the sequence for, say, green fluorescent protein, as shown in this example. So scientists found these bioluminescent jellyfish. They sequenced the gene for this fluorescent protein, and all they had to do was take that genetic sequence, the DNA sequence, and insert that sequence of DNA into these pigs to make them glow green. Right? So it's not like they had to find and isolate protein from the jellyfish and inject it into the pigs. No, they actually took the DNA that codes for that protein, put it into the DNA of these pigs, and it looks like in these pigs that the majority of the cells in their body have this DNA sequence, this gene, and the cells in that pig with its own transcription and translation machinery can read that code and make the exact same protein, that green fluorescent protein that the jellyfish makes. And you can see this pretty cool picture at the end of the glowing pig, all right? And so again, this code is universal, and that's what makes it so exciting. All right. This is also really good evidence for the idea that everything has a common ancestry, because why, if everything comes from a common original species, original genome, there's no need to be changing up what the code is, right? And again, it's also why we have such amazing advances in genetic engineering. Now, here we have a diagram of the actual process of translation. We've got our message RNA in this cyan kind of color. I don't even know. No, teal maybe? It's kind of a sad color. And then we have our ribosome in green. The ribosome's always green. And we've got our amino acids in pink. All right, so we've got a growing polypeptide chain. We've got an amino acid, phenylalanine, coming in that's going to be added to that chain of that growing protein. Now we said that the ribosome is what really catalyzes the formation of protein from this message RNA. So the message RNA is acting like a blueprint, right? And the way that you can take a genetic sequence, a nucleic acid sequence, and turn it into a protein, an amino acid, is this important RNA called tRNA. This stands for transfer RNA. So the transfer RNA is really neat because on one side it has an attachment site for an amino acid, and then on the other side it has a sequence of RNA. And so you can see here in this sequence of message RNA, we have AAA. If you look at the table, you can see that AAA codes for lysine. And so the transfer RNA for lysine has the complement of AAA in its sequence, which is UUU. And so what happens is all these different transfer RNAs are coming and going, and they try and see, is this the correct complement, right? Kind of like, does this Cinderella's slipper fit my feet, right? So if it's complementary, it stays. If it's not, it just leaves that site. Right? Similarly, the next sequence we have is GAU on our message RNA. GAU codes for aspartate, and so there is a different transfer RNA that only binds to aspartate and has this sequence on it, which is complementary, CUA. You can see coming in next the newest amino acid of the peptide, phenylalanine. And so this AAG sequence in the transfer RNA is called the anticodon. Right? The anticodon, it's the opposite, it's the complement to the codon of the message RNA. So again, transfer RNA, tRNA, it's another type of RNA. Right? Here we've got some different ways that people represent or draw tRNA. We've got kind of the cloverleaf model here. You can see on the acceptor end, that is where the amino acid binds. And then we have the anticodon loop. That is the sequence that is complementary to the codon in the message RNA. Now, the ribosome itself is pretty cool. It is actually also made out of mostly RNA, which is pretty cool, rRNA. And its two main functions, first, are to decode the message RNA, and second, to form peptide bonds. Right? So if you are trying to add amino acids together to make a chain, you have to form peptide bonds. If you remember way back from the beginning of the semester, forming these peptide bonds, that's a condensation reaction. Right? So in between each of these amino acids is a peptide bond. And so the part of the ribosome that forms this peptide bond is called a peptidyltransferase. And again, like I said before, the majority of the ribosome is actually made out of RNA, rRNA. Here we can look into a ribosome during translation. You should know that there are three binding sites for tRNA, A, P, and E. All right, so the first place where tRNA binds is at the A site. Then it moves to the P site and finally the E site. So tRNA comes in bearing an amino acid and comes into the A site and checks to see if it is complementary to the message RNA. If so, it binds and the ribosome moves forward. So it's going to move to the right, and this tRNA is going to be shifted to the P site. Now, what happens at the P site is that the tRNA in the P site is attached to the polypeptide chain. So the protein that's already been synthesized so far is attached at the P site. And then at the E site, this is where the tRNA sits that has lost its amino acid and it's about to be ejected from the ribosome. Now, I encourage you to find your own video, look up a video about translation if you didn't like the previous ones, because it's really important for you to understand what's happening at each site. So what I'd like you to focus on here is we're kind of going from left to right, top to bottom, and we're just looking at the various steps of translation. We're looking at the very beginning of a protein to be synthesized, and so you can see that at the beginning of the message RNA sequence we have AUG, and so a tRNA has already come in carrying the methionine and has bound and it's currently at the P site, and a transfer RNA containing a serine amino acid has just entered and bound at the A site. So this tRNA came in, the anticodon was complementary to the sequence UCC, and it's brought in an amino acid serine, and serine needs to be attached to the methionine. Now, the ribosome, we said, has this enzymatic activity. It's going to catalyze the formation of a peptide bond between these two amino acids. You can see now that this has happened, the tRNA at the P site no longer has its amino acid attached to it, and what's going to happen is the ribosome is going to move forward. So now the empty tRNA is in the exit site. The tRNA containing the polypeptide sequence is in the P site, and now the A site is ready to go for another tRNA to come in. So as you can see in step 4, that empty tRNA leaves the ribosome, and AAG codes for lysine, and so a transfer RNA that has a lysine attached to it is going to come in. It's complementary to the sequence, and so it stays in the A site. Next, what happens? The ribosome is going to catalyze a peptide bond between the serine and the lysine. And now once that's happened in step 6, you can predict what would happen next. The ribosome is going to move forward, that empty tRNA is going to be expelled, and a new transfer RNA containing an amino acid can come in and bind to that complementary sequence. If you remember, we talked about the 5' cap. This 5' cap is really important in helping the ribosome find message RNA that needs to be translated. And again, as we said before, the first amino acid is always methionine. And so this methionine is actually bound by an initiator tRNA. I don't expect you to memorize this, but it's just a little bit more detail of what happens at the very beginning. You can see that the ribosome actually has 2 parts. And so at the initiation of translation, the smaller subunit will come in, it will identify that 5' cap, and then the complementary sequence to AUG codes for methionine. So the methionine initiator tRNA will bind, and that basically is a flag to help tell the ribosome to come, the large ribosomal subunit to then attach. If you remember before, I said that the genetic code is degenerate. In other words, there is more than one codon that matches with a single amino acid. For example, leucine, this amino acid has 4 different codons, 4 different sequences that will all code for that single amino acid. And this is due to something called wobble pairing, right? So if you notice when you look at all of these sequences, they all start with CU, and the only thing that is different is the 3rd base in that codon. And so you can have a tRNA that is bound to leucine that has the anticodon GAG. And because of this wobble pairing, what can happen is that this anticodon GAG, while its perfect complement is CUC, it will also in a pinch bind to CUU. Right? So this 3rd nucleotide, this 3rd base, is not essential for the transfer RNA to successfully bind to the message RNA. And so you can have the same transfer RNA bind to 4 different sequences, as we see here. This is less stringent, it's less strict, right, at following the rules, and it's called wobble pairing. Again, just makes things more efficient. At the very end of translation, the ribosome knows to stop reading the sequence when it gets to UAA, UAG, or UGA. These are stop sequences. And they don't actually code for an amino acid or a tRNA. Instead, what they code for is a release factor. So a release factor will come in and it will bind to these sequences. And the ribosome identifies that release factor. It says, okay, I'm done. The ribosome lets go of the polypeptide that has just been made and separates in 2 and leaves the message RNA. It all falls apart. So again, we have an overview of transcription, translation, of the central dogma here. So transcription is going to occur in the nucleus in eukaryotes. The enzyme essential for this is RNA polymerase II, and it is taking DNA and using it as a template to synthesize RNA, a single strand of RNA. Before this single strand of RNA can leave the nucleus to undergo translation, it has to be processed. We have the 5' cap, the poly A tail, and splicing of introns. Once this has all happened, the RNA is able to leave the nucleus, travels to ribosomes, where the ribosomes catalyze the formation of a protein. Let's do a problem. For this sequence of message RNA, what is the anticodon and what is the amino acid that's going to be added to this polypeptide? So First of all, we have to figure out what is the anticodon. We know that the codon is a set of 3 bases in the message RNA. And so the anticodon is the complement of a codon, and that is found in the tRNA, the transfer RNA. The transfer RNA is what brings in the new amino acid. So if we know the codon, we can figure out its complement. We can also figure out using the genetic code what the amino acid is. So the codon that we have here is AGU. That means that its anticodon is UCA, right? It's just the complement. It's made out of RNA. And we know that AGU, if we look in the genetic code table, that codes for serine. So the next amino acid in this chain is serine. With the last objective, we're going to talk about mutations. Right? So before we start, do mutations say you are exposed to UV rays, right? What is being mutated? DNA, RNA, or protein? It's a tricky one, right? The actual molecule that first gets changed during a mutation event is DNA, right? And so if DNA is the blueprint, it's the genetic code, it's going to have a ripple effect downstream and is going to cause changes in the message RNA and the protein, right? So all of these mutations are happening at the DNA level. Mutations are basically changes in genetic sequence, right? So you can have a point mutation. This is when a single base in a gene gets changed. Say an A gets changed to a T, all right? You can also have a point mutation in which a base is just lost. Right? When you have a point mutation where one base is changed out for another, that is called a base substitution. Now you might think, one base out of, a sequence of thousands of bases, does that really matter? Well, let's think about what we know from this last semester. We know that structure determines function, right? So could a change in sequence change the structure of a protein and change the function of that protein? Potentially, yes. So we know that only 3 bases are needed to code for an amino acid, and if one of them changes, it could potentially result in a different amino acid being put into that protein. And we know that these amino acids are very critical for things in proteins such as active sites and binding sites, and so maybe an enzyme can't catalyze a reaction anymore. Maybe it can't bind to another protein like it normally could due to that one base change, right? Due to that one amino acid change. So when we have a mutation in DNA, we can have 3 effects when we have a base substitution, 3 results. You could have a silent mutation, a missense mutation, or a nonsense mutation. And there's an activity associated with this lecture, and you're going to be given some chances to kind of play around and compare and contrast these different mutations. So a silent mutation would result in no change, right? So we talked about how there are 4 different codons, for example, that code for leucine. Say you originally had a sequence for one of those codons and it changed to another codon that also codes for leucine. There was a change that happened at the DNA level and the message RNA level, but it did not result in changing the actual amino acid sequence, so it's called a silent mutation. A missense mutation results in a completely different amino acid being inserted, right? So maybe instead of a leucine, now it codes for valine. Then lastly, a nonsense mutation, this is the worst, it gets changed to a stop codon. You remember there's 3 different codon sequences that will tell the ribosome to stop making protein. And so if the mutation causes a sequence to become a stop codon, now what you're going to have is just this little stubby truncated protein that probably can't do much, right? That's a nonsense mutation. A third type of mutation I'd like to describe is called a frame shift mutation. So in this mutation, the addition or deletion of a single base causes the reading sequence of codons to be shifted by a letter. So this is going to have a really large impact because every single amino acid after this mutation is going to be different. I'd like to show you an example. Here we have a sequence, or a short sequence, from earlier in the lecture of DNA, its corresponding message RNA, and the protein that is synthesized. Now, if, for example, the fourth base in the sequence of DNA, that T, is lost, what happens is the ribosome is going to go on reading 3 bases at a time. And so the whole reading frame shifts, right? So the reading frame is basically the pattern, the set of 3 bases that it reads at a time. And so now what's going to happen is instead of getting AGA, which codes for arginine, the ribosome reads GAU, which codes for aspartate. And also the next, any of the following bases after that, the codons have also changed. And so this protein is going to look completely different after this mutation site. Another example that I want to show you is something seen in Huntington's disease. So in Huntington's disease, there's a mutation where a sequence of 3 bases, a triplet, get added over and over and over in a row, right? So you just have these 3 bases over and over, and what happens is that is really difficult for the ribosome to read and for polymerases to read as well. So for example, during DNA replication, we have the DNA polymerase, right? And it's going through and it's copying everything to make a copy of the DNA. Well, if you were, say, trying to copy a really long number and you had 5 zeros in a row, you might every once in a while lose track which 0 you were on, right? And so the same thing in Huntington's disease. The DNA is much more likely to get mutated, accidentally copied incorrectly, and then that can result also in a frame shift mutation during translation. So let's try and come back to connecting DNA with phenotypes, with natural selection that we've been talking about recently. So mutations are the starting point for evolution. If you remember when we talked about the different types, different mechanisms of evolution, we talked about mutation being one of those mechanisms. It is the ultimate starting point. It's rather infrequent, but it is the source of diversity, right? So we talked about how for natural selection to occur, the first thing that must happen is there must be variety in the population. And that variety only comes because of mutation. And so you can have mutations that are beneficial, right? Say you have maybe an expanded capacity of an enzyme to function. Maybe you have a mutation that results in a bird with more colors or spots, right? But it could also be harmful. We looked at some examples here in which the whole protein could be shut down, right? Not even made. And so you're going to have a lot of the negative mutations that cause an animal to die or to not be as fit are not going to be passed down to future generations because those animals don't get as many chances to reproduce. However, mutations that cause changes that are beneficial to the cell, to the organism, we're going to allow for more reproductive opportunities and for those genes to continue on down into the next generation and to eventually become very prevalent in that population. So in humans, we have found that Mutations occur relatively infrequently. However, one study suggests that there are about 70 new nucleotide changes in every person that's born. Okay, if you think about 70 out of how many bases there are in our entire genome, right? That's pretty good protection of our DNA. Even less rarely than a single nucleotide change is a insertion or deletion of like a fragment of DNA, right? And then even larger deletions occur or duplications occur even more infrequently. However, right, they do happen, right? So the single nucleotide base mutations, those are often used to identify individuals that are using forensic pathology, right? So they look at and see each person has their own kind of pattern of single nucleotide mutations, and they can use those to identify criminals and victims of crimes if they have any biological material. Now let's get back to talking about how genetics are connected and mutations are connected to evolution. You watched a video in lab about the rock pocket mice. I always tend to think of them as the rocket pocket mice. That's not quite true. So again, they're called pocket mice because they can shove food into their cheeks like pockets, and they live on rocks. And in the video, they talked about how there was variation in this population of pocket mice. Some were more of a sandy, a lighter color, some were darker, and depending on where they lived, there were selective pressures for the lighter or the darker mice. So when they were living in volcanic areas where the surroundings were darker, they were able to camouflage if they had darker fur. And the ones that couldn't with a lighter fur got eaten by predators. And so over time, in that population, it changed so that the majority of those mice had dark fur. And so the ability to even have variation in color is due to mutation, right? So a long, long time ago, there was only one color, right? There was no variation. There was just sandy colored mice, right? And so natural selection for this specific trait was not occurring because, as we talked about in the rules of natural selection, there has to be variability in the population, right? So no one had an advantage over the other because they're all the same color. The gene responsible for the fur color, or one of the genes responsible for fur color, is MC1R, as they talked about in the video. And the original sequence codes for the sandy color, the lighter color. Now, at some point in the history of this species, mutation occurred in this gene. They found several sites where there were mutations, and I actually looked up some of those sites. So the sequence of the gene is very long, so I'm not going to show the entire sequence of the MC1R gene, but I'm just going to show you little bits of it that were changed. So for example, there was a region, a codon that was GCG, and it got mutated to ACG. What this resulted in was a change in the protein that was made. GCG, the DNA, is what codes for arginine, and it got changed to ACG, which instead codes for cysteine. And so we have some of these other examples here, right? And so these mutations, they cause changes in the protein sequence, changes in the structure. We said that structure determines function. So this protein coded by the MC1R gene functioned differently because of these mutations. And the result of these mutations on this protein was that those cells had a different level of pigment, and so these mice resulted in having dark fur color. So this mutation caused diversity, right? So this mutation is an allele. This is where we get this idea of alleles, right? So the original sequence for that gene is the normal or wild type allele. This mutant allele is what's providing the dark color fur. And who knows, maybe down the line there'll be another mutation event and we will have a third allele for this gene, right? So again, this is how you can see that these physical characteristics, such as fur color, are due to changes at the cellular level, how pigment is made by these proteins, right? So this protein MC1R, right? It's coded by the gene MC1R. And so if there is a mutation in the gene that's going to change the activity of proteins, that will cause cells to change their activity, their functions, which on a larger scale can affect how that organism behaves, how it looks, how it appears, right? How it is able to survive and live in different environments. And so these changes in genotypes over time are due to selection of these different alleles depending on what is the most supportive for survival in that environment. All right. I hope you've enjoyed at least some of this semester. I have enjoyed getting to know you during the first half. Please feel free to email me with any questions that you have. I try to respond quickly. And best of luck on the final mini exam and your summer.